Hello there, I'm Felix,
And I'm John.
and today we will be giving a brief history of antisense oligonucleotides as a therapy for Duchenne muscular dystrophy. So first off, what is Duchenne muscular dystrophy? Duchenne muscular dystrophy, or DMD for short, is an X-linked genetic disorder that results in the loss of a protein called dystrophin. The dystrophin gene is the largest gene in the human DNA and codes for dystrophin, which is localized to muscle cell membranes. Dystrophin provides structural stability between the intracellular filaments and extracellular matrix, serving to stabilize the muscle as it undergoes contraction. More specifically, dystrophin attaches to actin and anchors the myofiber to the extracellular matrix via the dystroglycan complex. Without dystrophin, the entire dystroglycan complex disappears and muscles lose structural stability, resulting in tears in the muscle membrane. Tears in the muscle membrane result in progressive muscle wasting as muscle cells slowly lose their ability to regenerate. As muscles die, adipose and scar tissue replace degenerated muscle, and muscles eventually stiffen to the point where movement is severely impaired. Eventually, the loss of dystrophin leads to cardiomyopathy and a decreased capacity for ventilation, leading to cardiac and respiratory failure. This marks end-stage DMD and accounts for the staggering 26-year average lifespan. Before we talk about current therapies for DMD, we must first understand the cause for the lack of dystrophin protein on a molecular level. Similar to words in a book, our DNA is organized into three-letter words composed of DNA nucleotides. Each word represents an amino acid, and a complete sentence is equal to a complete functional protein. DNA is transcribed into mRNA, and the mRNA leaves the nucleus to be translated into a protein. DNA is composed of 3 billion base pairs and is divided into introns and exons. Introns are non-coding regions and do not code for a protein, while exons are coding regions which do code for a protein. When DNA is transcribed to mRNA, introns are spliced out of pre-mRNA sequence by a complex protein called a spliceosome. Once introns are removed, the now mature RNA can be translated into a protein. Here is an exon map of the dystrophin protein. As you can see, the exons are not identical and also seem to fit like a puzzle in some areas, such as between exon 50 and 51. These puzzle-like pieces represent areas where the three-letter sequence that codes for an amino acid or codon is split between two exons. This is where the most common cause of DMD lies. In DMD, a deletion of exons results in a loss of reading frame and consequently a dysfunctional dystrophin protein. In other words, the three-letter words in the sentence are shifted and the message is lost.
So how do we fix a mutation that causes a loss of reading frame? One potential answer could lie in exon skipping, which seeks to restore the reading frame in mRNA. A morpholino oligomer has a molecular structure very similar to that of nucleotides found in DNA and RNA molecules. Janet Heisman was one of the first to pioneer the use of morpholinos for gene knockdowns in vertebrate embryos. One of the first vertebrates she studied was the xenopus, which has paved the way for modern understanding of antisense oligonucleotides, or AONs for short. A morpholino is a type of AON and has DNA bases attached to a backbone of morpholine rings linked through phosphorodiamidate groups. Because of the similarity of the two structures, morpholinos can attach complementarily to specific sequences of pre-mRNA. When bonded, morpholinos have been proven useful in blocking other molecules, such as the spliceosome, from accessing the bound sequences of RNA. This mechanism has made them a particularly useful tool for exon skipping therapies which seek to restore the reading frame. There are three main mechanisms by which antisense oligonucleotides function. The first is through mediated degradation of an mRNA sequence of interest. Another is through the blocking of translation of the mRNA prior to entering the ribosome. The last involves the excising of pre-mRNA sequences unconducive to an open reading frame, and is the main mechanism by which current drugs such as eteplirsen are employing to treat DMD. Specifically, it prevents the inclusion of exons that do not lie in frame with the mutated sequence of mRNA. In other words, AONs allow for the skipping of unnecessary exons to produce a functional, albeit shorter, isoform of dystrophin that may still rescue sarcolemma stability, which would ideally result in a pathology similar to a less severe form of dystrophy known as Becker's muscular dystrophy. Like other morpholinos, eteplirsen has a structure that follows the same basic composition seen in nucleotides, including a phosphate sugar backbone with base binding sites. Developed by Sarepta Therapeutics, Eteplirsen was approved by the U.S. Food and Drug Administration in September 2016, making it the first and currently only FDA-approved drug for DMD. In one of their clinical trials, 13 patients were treated with eteplirsen, nicknamed Exondys 51, weekly for 48 weeks. Using Western blot analysis, dystrophin levels in muscle tissue were assessed at baseline and after 48 weeks of treatment. Many patients did experience a change in dystrophin levels from baseline, but the results remained insignificant. Sarepta is currently recruiting for additional clinical trials to demonstrate stronger evidence for clinical benefit. Although it remains at the forefront as a defense against DMD progression, at best the action of eteplirsen still cannot be deemed sufficient enough to satisfy the clinical endpoint of the trial. A cure for DMD is still not found, but active research is still underway investigating potential therapies. Currently, AAV vectors, CRISPR-Cas9 gene editing, and stem cell transplantation are other potential therapies that have shown significant results in animal models. Hopefully within the coming years, a cure for this life-threatening disease can be found. Thank you for listening. We hope you enjoyed this presentation.